Hi, welcome to EasyLearn 360. I'm really sorry for delay in posting videos continuously as I had exams. So this is the continuation of my previous topics, that is tissues. In this video, I am going to discuss about the classification of epithelial tissues. As we have already discussed in previously, that the epithelium is seen in our body in two patterns, that is as surface epithelium and glandular epithelium. The surface epithelium is again classified according to the arrangement of cells and the shape of the cells, and glandular epithelium is also divided according to the number of cells or the number of branches or the type of secretory, the shape of the secretory part, etc. So we can see in detail about the classification. So first we are going to deal with the surface epithelium classification. The surface epithelium is classified based on the arrangement of cells and shapes of cells. Arrangement of cells. In the previous videos, I have already discussed that the cells are arranged over a basement membrane, and that the top surface it is called apical surface and the side surfaces are called lateral surface. So based on the arrangement of cells, it is called a simple layer. Simple epithelium means that is the cells are arranged in a single layer, while in stratified the cells are arranged in one or two layers, one or more than two layers. Then pseudo stratified means actually it is simple epithelium. They are having only a single layer of cells, but due to the arrangement of nuclei in different levels, we may feel that it is stratified. So when viewed under microscope, it can give us an appearance of stratified epithelium. So these are about the arrangement of cells. Next, the criteria is shapes of cells. If the shape of the cells are square, then it is called as squamous. Cubes, cuboidal. Columns, columnar. And if these shape of the cells are varying or changing according to the elasticity of the uh, lining of that epithelium, it is called transitional. Transitional means changing. So I will explain this with a figure. So in this figure you can see that this is the basement membrane and the cells are arranged in a single layer. So this is simple epithelium. This stratified means they have more than one layer, and the above layer it is the surface will be called as apical surface, and this below one will be referred to as basal surface. In pseudostratified epithelium, unlike these two, the nuclei are arranged in different levels, so we get an appearance of a stratified epithelium. Actually it is simple epithelium, so it will be referred as pseudo means false. Then coming to the classification according to the shape. So here it is squamous means square shaped, cube shaped, column shaped. And in some cases, these cells will have small projections called microvilli or cilia. These are two different types of projections. Cilia means they can move particles over its surfaces, while microvilli they increases the surface area of that particular lining and it helps in absorption. So these are the two types of projections which are found over the cells, and the cuboidal cell, especially these are found in columnar epithelium, and these can again give the classification as ciliated and non-ciliated. So these are the criterias. So combining all these things, as a whole, the surface epithelium is classified as simple squamous, simple cuboidal, or simple columnar. If it is stratified, it is stratified squamous, stratified cuboidal, stratified columnar, and we have one more classification, that is transitional classification. Especially in columnar cells, they have microscopic cytoplasmic projections called cilia and microvilli. So this is cilia. Cilia are projections which can move the particles over its surface. So these are kind of protection, or in some cases it helps in the movement. In nasal cavity, these serves as a function of protection because it prevents the entry of foreign particles into the respiratory tract. while in fallopian tube, the cilia will help the movement of oocytes towards the uterus from the fallopian tube. The movement, it facilitates the movement of oocytes. And another one you should know about is the cells, goblet cells. Goblet cells are the cells which holds the secretions and releases the secretions. These are especially found in gastrointestinal tract. When these goblet cells, the mucus is getting filled inside these cells, these are released as secretions. So going over to the classification again. Simple epithelium, stratified epithelium, simple squamous, simple cuboidal, simple columnar, again classified as ciliated simple columnar, non-ciliated, pseudo-stratified columnar epithelium divided into ciliated and non-ciliated, stratified epithelium as stratified squamous, stratified cuboidal, stratified columnar, and transitional epithelium. And it is very important to know the locations of these epithelium, and you will come across all these things when you study each organs in details in anatomy. So when you hear about simple squamous, you have to imagine that these are single layer of squamous cell arranged in a one layer. So what will be its main function? It is a simple single layer. So it will serve the function of secretion or absorption or diffusion. So these will be mainly found in the linings of cardiovascular system and lymphatic system, usually found in these areas as serous membrane also, and is commonly referred to as endothelium, mesothelium, depending upon its location. And the main function of simple squamous epithelium is filtration, diffusion, secretion, etc. The next is simple cuboidal epithelium. These are single layer of cube-shaped cells, centrally located nucleus, and these are usually found on the surface of the ovary, lens of the eyes, kidney tubules, and smaller ducts of many glands, and makes up the secretory portion of some glands. Functions are secretion and absorption. Then coming to the columnar epithelium, it can be either ciliated or non-ciliated. In ciliated simple columnar epithelium, these are single layer of ciliated column-like cells with oval nuclei near the base of the cells, and goblet cells are interspersed among the ciliated columnar epithelium. These are found on bronchioles, fallopian tubes, uterus, some paranasal sinuses, central canal of spinal cord, and ventricle of the brain. So the function is cilia beat in unison, moving the mucus and foreign particles. Then non-ciliated columnar epithelium, here they have no cilia over its surface, and they may have microvilli on their top. And goblet cells too, they have goblet cells and microvilli. Microvilli increase the surface area and they increase the rate of absorption. And goblet cells are modified cuboidal cells which will secrete the secretions according to the location. Usually they secrete mucus. The next is pseudo-stratified columnar epithelium. It is again classified as ciliated and non-ciliated. Ciliated pseudo-stratified columnar epithelium have cilia and goblet cells. They line the airways and most upper respiratory tract because we need cilia to push away the foreign particles. So remember like that. So these are found in the respiratory tract. The ciliated simple columnar epithelium also, they are located in the nasal cavity. So we can correlate the function and then memorize the type of epithelium. Okay, coming back. Ciliated pseudostratified epithelium, these are found on the airways of most upper respiratory tract, and they secrete mucus that traps foreign particles, and the cilia sweep away the mucus for eventual elimination from the body. Then the non-ciliated pseudostratified epithelium, they appear to have several layers because of the placement of the nuclei at various levels, and though the cells are attached to the basement membrane, some cells do not reach the apical surface. They lack cilia and also they lack goblet cells. So they are found in the lining of epididymis, in the male reproductive organ, then larger ducts of many glands such as the parotid glands and parts of male urethra. So these are about the simple epithelium. Then coming to the stratified epithelium, stratified squamous epithelium. That means It is the epithelium with square shaped cells which is arranged in different layers. So these are usually found in the epidermis of our skin. So as you know, in the skin has a function of protection. So for protection, they need many layers. So we can think like that. So in case of skin or epidermis, or in case of stratified squamous epithelium, they consist of two or more layers of cells, and the cells in the apical layer and several layers deep to it are squamous. But cells in the deeper layers, that is near to the basement membrane, they appear columnar. As basal cells divide, cells arising from the cell divisions push upward toward the apical layer. And as they move towards the surface and away from the basal layer, they may become dehydrated and less metabolically active as they shrink in size. So, as their cytoplasm is reduced, the percentage of tough proteins called keratin predominates within the cell and they become tough. So, the stratified squamous epithelium we can again divide as keratinized and non-keratinized, depending upon the location. The next is stratified cuboidal epithelium. Stratified cuboidal epithelium, this is a rare type of epithelium found on the adult sweat glands as well as esophageal glands and part of male urethra. These are again the cube-shaped cells arranged in different layers. Again here also the function is protection and limited secretion and absorption. Mainly these are for protection. Then next is stratified columnar epithelium. Stratified columnar epithelium, usually the basal layer consists of shortened irregularly shaped cells. Only the apical layers have cells that are columnar in shapes. So these are usually have the function of protection and secretion and are found on part of urethra, large excretory ducts of some glands, esophageal glands, and conjunctiva of the eye. Then next is the transitional epithelium I have already discussed. These are having different shape based on the relaxation of that particular organ. In its relaxed or unstretched state, transitional epithelium looks like stratified cuboidal epithelium, except that the cells in the apical layer tends to be large and rounded. As the tissue is stretched, the cells become flatter, giving the appearance of stratified squamous epithelium. Because of its multiple layers and elasticity, it is ideal for lining hollow structures that are subject to expansion from within. So these are usually located in the urinary bladder and portions of ureters and urethra. Functions are it allows the urinary organs to stretch and hold a variable amount of fluid without rupturing while still serving as a protective lining. So these are about the classification of surface epithelium. The second main classification of epithelium is glandular epithelium. That is, they are found as glands which secrete secretions or hormones. So what are glands? A gland consists of epithelium that secretes substances. So what are the two ways of secretions that you should know? That is, either they are secreted into ducts. That ducts are small, hollow, tube-like structures through which the secretions will come out. So these are ducts. So sometimes these secretions are secreted directly into the bloodstream. So these are the two mechanisms by which the secretions are secreted out from glands, either through ducts or directly into the bloodstream. If it is secreted through a duct, it is called exocrine glands. And if it is excreted directly to the bloodstream, it is called endocrine glands. And such secretions are called hormones. And these endocrine secretions, they can travel through the bloodstream and can act on organs which are away from its site of production. While in exocrine glands, these are excreted through, they are secreted through ducts. So they can act on the primary site of production only. So these are the basic difference between endocrine gland and exocrine glands. Endocrine glands example are pituitary, thyroid glands, etc. And exocrine glands example are pseudoriferous glands, that is sweat glands, sebaceous glands. All these are examples for exocrine. Then we have classification of glandular epithelium based on the structure as well as based on the function. Based on the structure, it can be classified either as unicellular or multicellular. Unicellular means they are single-celled glands. Example, goblet cell. Only that single cell will produce a produce a secretion and will secrete it. Goblet cells. While multicellular means they have many cells. It can be either microscopic or it can be macroscopic organ. Example, pseudoriferous, sebaceous gland and salivary glands. And another classification is based on the branches. If it is having no branch, it is called simple gland. And if it is having branches, it is called compound gland. So simple and compound glands. So the exocrine glands can be classified as unicellular or multicellular, and based on the branch it can be classified as either simple gland or compound gland. Based on the shape of the secretory portion of the gland, it can be either tubular or rounder, or it can have both of those. If it is tubular, it is called tubular gland. If it is rounded, it can be called as acinar or alveolar glands. And if it is having both, it is called as tubulo-acinar glands. So the final classification is like this. Simple glands, it can be simple tubular, simple branched tubular, simple coiled tubular, simple acinar, simple branched acinar. This branching is different from this one. I will show in the figure. Then compound glands, it can be compound tubular, compound acinar and compound tubular acinar. This figure, you can see that it is the simple gland means it is having only one duct, main duct. It is not dividing. Here also, this main duct is not getting divided. But here you can see that it is divided, and that divided gland is being rounded or tubular. So these are the basic difference between simple and compound. And here it is simple tubular, that is it is having no branch and it is tubular in structure. Here also it is tubular but coiled. Here it is simple because it is having only one main duct but branched. So simple branched. Then here it is compound, that is the main duct is being divided into three and it is tubular. Here it is rounded. The secretion portion is round in shape. So it is called compound alveolar or compound acinar gland. So these locations of each, that is simple tubular glands, these are glands which are found in large intestine. Simple coiled glands, these are sweat glands. And simple acinar glands, this one. Simple acinar glands, these are glands of the penile urethra. Simple branched acinar, these are found as sebaceous glands. And simple branched tubular, these are found in gastric glands. So this one, large intestine, sweat glands, gastric glands, and this one is about penile urethra, and this one is sebaceous glands. The next is compound glands. Compound tubular, example is bulbourethral glands or Cooper's gland. And compound acinar glands, these are typical of mammary glands. And another classification is which is having both tubular and acinar end. So these are found in the pancreas, acinar glands of pancreas. So this is about the structural classification of glands. Now based on the functional classification of glandular epithelium, that is the mechanism by which these secretions are secreted out. Based on that, the exocrine glands are divided into three, that is eccrine glands, apocrine glands, and holocrine glands. So in glands, imagine in a cell, these ribosomes synthesize secretions which enter endoplasmic reticulum, where they are processed, stored, and packaged by Golgi complex, and they are released from the cell in secretory vessels. In eccrine glands, these secretory vesicles reach to the surface of the cell, perform exocytosis, and they are released as secretory vesicles itself. In apocrine glands, what happens is that it will reach the surface and it will get pinched off from the surface. And in holocrine glands, what happens is that the secretions get filled up in the whole cell, and the whole cell will burst. So this is how the secretions are released. I will show in the figure. So this is the eccrine or mirocline gland. Here, this is a diagrammatic representation of a cell. This is a nucleus. This is endoplasmic. So from the ribosomes, they will synthesize and enter the endoplasmic reticulum, because the ribosomes are on the surface of endoplasmic reticulum. These secretions are stored, processed in the Golgi complex and packaged in the Golgi complex, and they are they are pinched off from the Golgi complex as secretory vesicles. And these vesicles, secretory vesicles, they reach the surface of the cell and they perform exocytosis and they are released. So this type of glands are called eccrine or mirocline gland. Most of the glands are eccrine or mirocline. The salivary glands, pancreas, everywhere saliva secrete like in this manner. Then next is apocrine glands. Apocrine gland means here the cell, the same mechanism, the secretory vesicles, they will reach the surface. No exocytosis is not happening. Instead, they come to the end and they are getting pinched off from the cell surface. And these are called apocrine glands, and these are usually found in the mammary glands. They are pinched off. The secretory vesicle as such is pinched off from the cell. Then the next is the holocrine glands. Here the holocrine glands, the secretions fill up the whole cell. And when the secretions reach to its maximum, the cell will rupture and these secretions are released. And these type of glands are called holocrine glands. Example, sebaceous gland. The sebum is released like this. So these are about the functional classification of glands: holocrine glands, apocrine glands, and eccrine. Eccrine, they are released by exocytosis. Apocrine means they are pinched off from the cell surface, and holocrine means the secretions, when they fill up, they are the cells rupture and the secretions are released. So this is all about the classification of epithelium, the surface epithelium and glandular epithelium. If you like this video, please like, share and subscribe my channel. Thank you.